Welcome to this special live and interactive Chemistry World webinar, where we're discussing the Nobel Prize in Chemistry 2022, which was announced on October the 5th. I'm Natalie Starkey, Chemistry World's science media producer, and I'm here for about the next hour to look at this year's prize-winning science and see how the world of chemistry has reacted. I'm joined today by some special guests from across the chemical sciences, who I'll introduce shortly. Firstly, I'd like to thank our sponsors, Shimadzu and Jump, who have enabled this webinar to happen. Now, the Nobel Prize announcements always cause a bit of a stir, and whether you're disappointed that your favourite area of chemistry didn't win this year, or delighted with the result, we're very glad you're here to join us. We welcome your thoughts on the announcement, and you can go to the GoToWebinar platform that we're on today to interact and ask your questions, or just share your comments and thoughts. We'd love to hear from you, and you're welcome to post questions at any time from now. As a quick explainer of how to do so, on GoToWebinar, there's usually a panel down one side of the screen, although this might be different depending on which platform you're using, especially if you've joined us via a tablet or phone. Now, somewhere on that panel, there's a questions box that you can use. A link to a recording of this webinar will be sent out to everyone who registered, and we'll also be uploading it to our YouTube channel. Now, before we begin and get stuck into the science, we'd really love to find out a little bit more about who our audience are today. So we're going to launch our first poll. And we're just basically asking you to tick the answer that best applies to you. I'll give you a couple of moments to do that. You can see we've got quite a lot of academic researchers in, which doesn't surprise me, and also quite a lot of students coming in. I'll give you a few, maybe another minute to put in your answers. Got still quite a lot of people voting. Thanks for interacting with us today. It's great to know you're all there and listening. Thank you all for voting. I think we can share the results because it might be quite interesting for people to see. There we go. So yes, as we can see, actually a really good mix of different types of audience here today, which I'm really excited about. Obviously, it shows how excited people have been, how we've all been excited about this Nobel Prize. Now, as you may be aware, the 2022 Chemistry Nobel was shared by 3 scientists this year: Carolyn Bertozzi, Morten Meldal, and Barry Sharpless, for the development of click and bioorthogonal chemistry. A huge congratulations to this brilliant team of scientists and all those involved in this research over the years, because as we know, there's a lot of teamwork involved in science. I just want to show you these pictures from Twitter that we found of 2 of those Nobel winners on the day they received that much coveted call from Sweden. Carolyn was actually awoken in the middle of the night, but that obviously hasn't dampened her excitement. And I love that Morten is celebrating with a mug of something in his office. I have no idea what might be in that mug. I'm sure their days were completely overwhelming. And in fact, what we have now is a little clip from one of our correspondents, Katrina Kramer, who spoke to Morten Meldal just this morning. And we've got a part of their conversation that you can listen to, where Morten is reacting to his call from the Nobel Committee. So let's have a listen.
Yes, I was sitting here preparing teaching material, so I record all our lectures on videos for the students, and I was actually doing that while, then I got the call. Unfortunately, I put my sound on the telephone because I normally don't have that. So I was very fortunate to actually get the call at 11, and then for 1 hour I was told I couldn't tell anybody, so I just kept quiet. Went over for a cup of coffee, and on the way I met our head of institute, and she was looking strangely at me because my smile was going from ear to ear, of course.
How hard was it to not tell her?
That was hard, but there wasn't that much time until they announced it. So soon as they announced it, the hallway outside my office was completely full with very enthusiastic students and colleagues, so that was very nice.
Yeah, I think I saw a video of you coming out of your office and people being very cheering, and that looked like a lot of fun. Who was the first person you told? Did you call anyone from your family or friends?
No, because as soon as this happened, the whole, 25 seconds after the announcement, the whole floor was full of people. So I didn't call anybody for a while. I actually was called by every 15 seconds there was a telephone calling me.
Isn't that lovely to hear? I can't even, having heard that, I still can't even imagine getting that call. And I know I will never be receiving that call, but it's just fantastic to get an insight into what these people have gone through. Now, I have a lovely set of guests joining me today to discuss this prize, and I'm excited to hear more from them soon, and we'll meet them all in just a minute. They're going to tell us about the people that have been awarded the prize and offer us some insights about the science in general and its applications. So the people we have are Philip Broadworth, who's a business editor at Chemistry World, and we have May Copsey, who's an executive editor of RSC Journals. We've got Alison Hume joining us from the University of Edinburgh, who actually uses click science in her laboratory. And we've got Nicholas Riley, who's currently a postdoctoral fellow in the Bertozzi group at Stanford, but he's also going to be incoming assistant professor of chemistry at the University of Washington in 2023. And we also have MG Finn from Georgia Tech, who actually worked on click science with Barry Sharpless. So we have a really exciting lineup for you. But actually first we're going to chat to Philip from Chemistry World, and he's going to be able to tell us a little bit more about the science that won this year's Chemistry Nobel. So welcome, Philip. Thanks for joining us.
Thanks for having me. Yeah, I mean, I think this is a fantastic prize. First of all, it's it's one that's been tipped for a little while. I think in the simplest sense, this prize is about kind of having ultimate control over chemical reactions. I did a PhD in synthetic organic chemistry myself, and the thing that you think about is a lot of the time, building molecules can be really hard. We're fighting against thermodynamics. We're fighting against trying to bend the physical universe to what we want it to be. And oftentimes that's messy. It's slow, it doesn't work very well. It works well enough, but there's side reactions or there's byproducts, or you have to spend time purifying things. And we kind of accepted that as the cost of making molecules, right? So but then click chemistry is, as I see it, it's a kind of a concept. It's about doing things a different way. Right,
and it's not just one reaction we're talking about here, is it? It's a kind of a whole group of reactions.
Yeah, absolutely. And I'm really excited to see what MG has to say about this later, because he was involved in kind of coming up with that whole concept. But the idea is that you're kind of, you want to use reactions that are clean and fast and high yielding and selective and mild, and build molecules in a completely kind of different way. And that's a great concept if you've got the reactions to do it. So then you've got a bit of pressure to kind of go, well, what reactions fit that bill? And it's quite a strict thing. And then when you find those reactions that work that way and they fit with that strategy, and you can develop them, and that's kind of, I guess, what Morten Meldal did and Barry Sharpless, and then taking it on. That's when it starts to become it goes from being an interesting concept to something that is going to potentially win you the Nobel Prize.
And what about bioorthogonal chemistry here? So how does that fit into this story?
Well, kind of the way I see it, and I'm sure Nicholas will probably correct me if I'm wrong, but bioorthogonal chemistry seems to me it's like the killer application for click reactions, right? You've got all this complex biochemistry going on in and around cells, and you want to be able to see what's going on. You want to be able to do things inside cells without disrupting that and explore it. So when you've got these kind of single-minded reactions, they will kind of go in and they can ignore everything else that's going on and just kind of click together, super high efficiency. That's really powerful. You've still got to do a huge amount of work to kind of, if you want to attach something to something that's in the cell already, you've got to have one partner of that click reaction, whatever it's going to be, attached to the biological system. So that was one of the key things I think that that Carolyn Bertozzi's group kind of worked out, was how to introduce one partner to the actual biological system and then use the chemistry to attach something that would allow you to see it. And then, building on from that, building and building and building, it's kind of exploded as an area. And I'm sure, all the rest of our guests today are going to expand on and tell us a bit more about how that's happened.
Definitely, I'm sure they are. And I'm wondering, with your business editor hat on, how, are these reactions used in industry? Do we know much about that at the moment?
Yeah, I mean, in a general sense, I am sure that there are medicinal chemistry medicinal chemists and people using this kind of chemistry, definitely using it in kind of assays and things. But if we look at, for example, Carolyn Bertozzi's group, she's spun out, I don't know, maybe 7, 8, 9 different spin outs from her group doing various different things using this kind of bioorthogonal chemistry, everything from protein degradation to treat cancer, to ways of detecting TB, ways of interacting with the biology of rare diseases, diagnostics to say, will a particular cancer drug work for a particular patient, all of those kind of things. And there's some really exciting things going on. I think Lysiothermal Therapeutics, which is one of the ones has the kind of cancer stuff, has a product in phase 1 clinical trials now. So that's, we're starting to get to the point where not only is this a useful tool for chemistry, it's having impact in pharmaceuticals, in products as well. And then you've got the whole kind of materials side of things. You click stuff onto different polymers, different things. So yeah, definitely has
just
impact
endless,
and application.
isn't it?
Yeah.
Definitely. Were you surprised by this result? Did you predict this?
Well, I'm not in the business of predicting it. It has been predicted by a lot of people who have been kind of looking at this thing for a while. It's been on the cards, put it that way. I mean, nobody ever really knows exactly what's going on behind the doors of the Nobel Foundation, but I think a lot of people would be unsurprised, and a lot of people are really excited because, I think all 3 of these prize winners are just fantastic chemists, really big members of the community, and really the reaction has been very, very happy. But
it's been so positive, hasn't it? It's been brilliant from what we've seen so far. I'm going to bring May Copsey in now, if she can turn on her webcam. Hi, May, welcome.
Thanks, Natalie.
So as someone who works in the world of science publishing, I'm just interested to see kind of how this kind of science has been developing over the years in the literature, since those initial first papers, kind of what's happened since then? Has it always been a big topic that you've been watching, or has it kind of grown over time?
Well, I'm obviously going back to sort of the beginning when, as you say, the Sharpless group were putting out this concept of click chemistry. We've gradually seen the number of papers and articles using click chemistry sort of just growing, and for at least the sort of past 10, 12 years, we've seen papers in the thousands each year being published using click chemistry. I just did a quick search across RSC journals, and I think over 1,000 RSC journal articles last year mentioned click chemistry. I mean, that's RSC journals alone. So that's just give you a sense of the sort of how click chemistry has really just embedded itself across so many different areas of the discipline. And to pick up a little bit on what Philip was saying, it's not just confined to sort of organic chemistry or medicinal chemistry or even chemical biology. We see this across a whole range of different journals in chemistry. Particularly the polymer community. I just want to call that out actually, because looking at the sort of the output in research across the different areas of chemistry, actually it's polymer chemists who've been really using this as a strategy in their sort of polymer synthesis to really great effect, and so yeah, it's really had a huge impact on a really large range of researchers.
That's quite amazing, and it completely makes sense. With that number of papers coming out every year on this topic, it's no wonder, it was deserving of a Nobel Prize. I mean, do you agree? Do you think it was these areas are deserving winners?
I mean, absolutely. I mean, it was when I was talking with some of my colleagues when it was announced, and several of them said, "Oh, I used click chemistry in my PhD," and you could just sort of see the delight that people feel when they can actually relate to the sort of the chemistry that's being recognised by the Nobel Prize. So I think that's sort of why there is such a lovely buzz about this one, is because I think people can really relate to it and and feel sort of connected to it somehow.
That's so great. And we're going to actually now bring on our other guests, and it's a good segue to speak to them. And these are the real scientists, some of the real scientists who are using these methods. So if we could have MG and Alison and Nick, if you'd like to turn on your webcams, I'll just wait for them to all appear. Hello, welcome to all of you. Thank you so much for joining us today from your various parts of the world. It's so good to have you here, and as I said, speak to people that are using these experiments and just all these methods. I'm really excited. I'm going to start with MG because as many people will be aware, you've been involved in click chemistry right from its very inception, in fact. When you go back to the original papers, you're there, and it's absolutely amazing. I'd love to hear your perspective on this. You must feel incredibly proud to have this science, which I guess you feel is your science as well, being recognised. And can you tell us a bit more about your link to Barry Sharpless, who was obviously one of the winners, and your involvement in this, what I'm going to call Nobel winning science now?
Well, it's just a delight. What a great day it was yesterday. We're all super excited. Philip, by the way, you did a great job describing this all, so well done. Yeah, so I was very fortunate to be a graduate student in Barry's lab these many years ago, and received my PhD under his direction. And then I went off on my independent career, and then got hired at Scripps back in the late 90s, and was a colleague of Barry's at that time. And so, again, just extraordinarily fortunate to be able to walk over to his office and shoot the breeze and work together and collaborate. And so I've just been the recipient of great good fortune to be able to work with him and learn from him. Just terrific. And of course, I know Carolyn very well, and Morten. They're just phenomenal scientists. This is just a terrific prize.
I like that you're basically referring to luck there, but I'm pretty sure it's not just luck. I'm sure there's been a huge amount of hard work that's gone into developing this whole side of click chemistry. When you first did these experiments, did you get the sense that it was going to be big? Was it easy? Did you think, oh, this is Nobel worthy work, or were you just kind of plugging away and not really sure?
No. It was so Barry discussed the idea with me very early on. He had been formulating it for a few years and arose out of a very organic thought process on his part. It didn't take me long to become convinced that this was a really cool idea. Although in Barry's signature fashion, he just decided to do it like on the spot, and pity the poor postdocs who come from Germany or wherever in the world to work with Professor Sharpless on, asymmetric osmium catalyzed dihydroxylation or something, and they arrive and Barry said, nope, that's not what we're doing. We're working on this crazy idea, and so it took a while for people to pick up on it. It was fairly obvious to me that it was going to be big if we could, as Philip correctly noted, discovering these reactions is really, to me, the quintessential. The epitome of chemistry, right? Good reactions are what it's all about. There aren't that many of them, as you pointed out, and discovering new ones are really hard. So I was not involved in the copper reaction discovery. I was involved in using it and studying it and figuring out how it works and applying it in a variety of ways. But that also arose in Barry's lab in a very organic way. He had his eye on azides for a long time, and in fact, we published before the copper reaction a very interesting use of azides and alkynes, which we called in situ click chemistry. And so azides this could be the Nobel Prize of azides, by the way. And Rolf Huisken and his, inspiring chemistry, and Carolyn was all over this, very early on with the Staudinger ligation, which was a really seminal contribution. So it was in the air. The power of selective chemistry was certainly in the air because, biological molecules and modifying them is always the goal. So anyway, there's a whole history here, but it was a very exciting time.
And how did the community react? Like, when you went to conferences and start presenting this work, how did you feel that community were reacting to it? Did they feel like it was something that was just, maybe not that exciting, or was there instant excitement about what you'd done?
No, there was quite a bit of pushback from organic chemists, a lot, because really, and I always understood that. Why do you feel the need to put a new phrase or a new title onto what is just really good chemistry? And that was a fair criticism in a sense, except that they didn't really understand what Barry was driving at. It wasn't the fact that these are good reactions. It was what comes when you relentlessly commit yourself to using as much as possible only those kinds of reactions. What benefit does that give you in discovery and production and the ability to, try to have as much control over chemistry as nature does with a 3 billion years head start? It was really the idea, the philosophy, as Philip alluded to, was really what was missing, what some people didn't pick up on right away. So there was a lot of pushback from the local community, but Barry didn't care, and the rest of us didn't care. We were just having too much fun.
That's amazing. I love that. Now you're running your own lab now. I'm guessing you're currently using click chemistry in your lab. I don't know. Is that something you're using all the time? Yep.
Every day. Yeah. Yep.
And what kind of work are you using it for at the minute?
So we do a lot of chemical biology, which is the field that Carolyn has really, led the way in. We use the copper reaction to attach high value compounds we do a lot of work with virus particles and develop vaccines and immunostimulatory reagents. So we do a lot of immunology, but it's all enabled by the ability to make bonds reliably. We also do a lot of material science. And as you correctly noted, the polymer community, we're really the first to pick up on the power of click chemistry in a real way, and we use that as well. So we use it every day. It's just an enabling thing. We don't have to worry a lot of times about connecting A to B, and that gives us the power to worry about why we want to connect A to B, and that's the real power of click chemistry.
What do you see as the future? I mean, it sounds to me like it's completely endless, like we can just apply this to any problem we want to apply it to. But what do you see as kind of the future trends with this science?
You can think about it in several ways. You can think about what's the future of click reactions, and that's really the future of, a one branch of reaction discovery. And there are certainly lots and lots of things to do there. The list of click reactions is rather small. And every time you develop a new one, you find a new one, or you resurrect a new one from the old German literature, it's a whole new world opens up. So there's that. I can steer people to an essay that Barry and Hartmut Kolb and I wrote in Nature Synthesis this year, in which we speculate about the future of click chemistry and what we're excited about. So people can read a much longer answer there than here. But what we landed on there really was focusing on the power of selectivity in a sense more than reaction rate, and the idea that we should be able to find, or we believe that the community will be able to find, chemical reagents that react with biological molecules in a way that it goes more than selectively in terms of chemical type, but really reaching out and reacting only to a particular structure or a particular residue at a particular pKa or whatever. The selectivity principle there, where you don't react with anything you don't want to, and you only react with what you want to, that's what's really at its infancy in the area of drug development and probing biological molecules. And so the more we can do things in vivo in that fashion, the faster the world will change.
And do you think this Nobel Prize is going to make any difference? Or do you think, I mean, it sounds like it's such a ubiquitous process that's used that maybe it's just very popular anyway, but does this kind of thing help bring students in and get people, younger scientists interested in the field? I'm guessing it does. It's already got me very interested in this new field I didn't know anything about before Wednesday.
I think so. One of the lovely things that we like to have students understand is that chemistry and biology intersect in ways that are not always obvious to the youngster. The Click Chemistry Prize, I hope, will allow young people to realize that they can, with knowledge and creativity in chemistry, go and do something in a biological context or in a complex context of the world, environment, materials, whatever. Powerful chemistry is the engine here, it enables so much. And to the extent that we can have young people realize that and realize the power of chemistry to do sophisticated things in sophisticated contexts as simply as possible, the more people of that mindset that we bring into the field, you know, the better we all are. Carolyn, I heard her give an interview, and she was talking about how excited she is for chemical biology to get a boost here from, you know, people who don't know anything about it. And yeah. I couldn't be more in agreement. And the other thing is, you know, new click reactions mean new materials, and new materials are always a new world. And that's my go-to when I think about the applications of click chemistry. In addition to the biology that we do, it's new materials in the world that have new functions. So
thank you so much.
Yeah,
that's
thank you.
We'll be getting some questions in from the audience. We're already getting quite a few questions in. People seem quite excited about this topic, which is great. I'm going to come to those audience questions shortly, but I'd like to just bring in Alison next, because Alison, I understand, you know, you use click chemistry. Once again, you're one of these scientists that uses this chemistry in your lab. And obviously we heard from May earlier, it's everywhere, it's in all the papers now, this is what people are doing. I'm just wondering, are these reactions easy to run? You know, can undergrads do this kind of stuff, or is it only, you know, advanced postdocs that can do this work?
No, I mean, it's really easy to use click chemistry. And I was just looking back to see when we first published using click chemistry. And actually it was like in 2006, 2007. So not that long after, you know, some of the initial reactions. And it was very obvious. It was very timely for us because we were looking at making small molecules, and my background is as a synthetic chemist, but I always wanted to know what the small molecules that I made, what they did. And so we wanted to track them inside cells. And it was really nice to be able to just put on a tiny little alkyne handle and then be able to click on a fluorophore or click on biotin to do pull down experiments, or, you know, so the reaction was, you know, the chemistry was very, very timely for us originally. I would say, because undergraduates, I thought that was, so undergrad students, I've had undergraduate project students do click reactions. That's absolutely fine. But you know what? Biologists can do this reaction, okay? They don't even have first year chemistry, all right? And that is literally it. I collaborate with so many biology labs, and they know what a click reaction is. And they buy kits, and they pay a lot of money to buy the kits. We won't say from whom, but they pay a lot of money to buy kits that have, you know, reagent A, reagent B, reagent C. They put the three of them in, and bang, they've got a fluorophore attached to their biomolecule or whatever it might be. You know, so it's that simple to do. I think that's the beauty of it. Absolutely.
So you don't even need all that, you know, that real chemistry background. You just use it. Just, you know, if you want to get more involved, sure. But if you just want to, okay, you're going to tell me off now, aren't you?
So yes, but no. Okay, because I think Philip alluded to the fact that so more recently, just at the moment, we're using, you know, the bioorthogonal chemistry. And for me, that's where the absolute beauty of the whole field is, the fact that you might take these reactions and do them inside a cell. And so we've hijacked Carolyn's, you know, glycobiology kind of stuff, and we're using it to put on, my group do a lot of work with Raman microscopy at the moment. So we're looking at ways to be able to track molecules by vibrational spectroscopy. And the term bioorthogonal is so brilliant for us because I talk about bioorthogonal spectroscopy because the alkyne bond is a part of the spectrum that has no cell vibrations. So we have a handle that's, you know, bioorthogonal. And so it's just such an amazing term to have come up with. But yeah, no, you know, if you want to do that, you've got to be a bit smarter because, you know, it's getting your handle onto a biomolecule in a living system that you can then go and do some chemistry with. You know, some of the advances in strain promoted click chemistry or whatever we might use, you know, that's quite a lot more complicated.
Okay, I'll trust you on that one. Yeah. Now I know that I think that you're using this technique for cancer kind of research and things. I've had a question in from Rajdeep about this actually. Has this technique been applied to cancer research yet? Which is a great question.
Well, so we might let Nick answer some of that, but we are also using it. So we are using it as a way of labeling T cells. So that we can look at tumor infiltration, because there are new T cell based cancer therapeutics that people don't really know how they work. They don't really know how the tumor cells. So one of the things that I'm very interested in is where small molecules go to, or where larger molecules go to, or where cells go to, and can we actually like visualize that in real time? So yes, we are using bioorthogonal click chemistry right now to track live T cells. Yeah.
Okay, I think that's a great segue to Nick, actually. So Nick, thank you again for joining us. Now, it's very exciting. I'm sure you're all very excited about this this prize because you work in Carolyn's lab. And so really, I'd like to come to you first just to say, what's it like working in a now Nobel winning prize lab, and what's it like to work with Carolyn? I've heard really only good things about her over the last few days. What does it take? Has she got a special thing about her that makes her a Nobel winning scientist? Yeah, if you can give us some insight into that, that would be amazing.
Absolutely. Well, it's a wonderful lab to be a postdoc in, to be a graduate student in. I've just had a wonderful time, and I've been in her lab for 4 years now. And I'll start by saying everything that you've heard about her and how great she is true. She is truly a wonderful mentor and person, and you see the jubilation for all 3 of the winners. I think it just underscores how much they've impacted the community, not just with their science, but with their presence and their ability to kind of establish the trainee aspect of how to spread these ideas. So being in her lab is, she is one of these people that cuts to the core of a problem in a way that I've never seen before. That's what I think one of her gifts as a scientist is. She sees there's levels of complexity, but she says, well, I can target in on what we actually need to solve, and let's be creative about how we solve that. And so that's what I see as her, like when she brought these ideas from the click chemistry world into the bioorthogonal chemistry idea of, we have these challenges for these really complex and hard to study biomolecules called glycans. How can we use these amazing chemical reactions that are selective and can really help us tune up our signal to noise so that we only focus on the biomolecules we want? She saw that and was, had all the background and almost reverence for the chemistry to bring it to the biological system she wanted to study. And so I think her ability to kind of cut to the core of a problem is something I've seen since day one, even interviewing for in her lab as a postdoc, talking about ideas of what I was interested in, where she thought I could contribute to the lab. She automatically was talking about ideas in the mass spectrometry world, which is my background, my training in graduate school as a mass spec person. She picked up on ideas incredibly quickly and like, oh yeah, I see how that could help. Or what if you did this in a field that she doesn't think that much about often? And so it's always been impressed upon me of how quickly she understands the core issue and then how creatively she approaches her problem solving to that.
No, that does sound amazing. So yeah, I think she's very deserving. A clever person, goodness me. This being able to connect all these things, it boggles my mind completely. So it'd be great to hear a little bit more about the work that you're doing actually in a bit more detail. What kind of applications are you doing at the moment?
Absolutely. Well, I'll share a little bit about my personal projects, but I think it'd be helpful to hear about some really exciting projects all around the lab. And I will say, as a mass spectrometrist, I get to kind of be involved in tangential ways to a lot of these characterization projects. And I've seen how beneficial click chemistry and bioorthogonal chemistry can be for an analytical chemist like me too. And so, and hopefully a growing chemical biologist, training with Carolyn. But the main focus of my work are mucin domain glycoproteins. These are really large proteins that exist on the cell surface, and they carry very dense regions of glycosylation. And so these are just sugars that are on proteins at the cell surface, and that affects how cells communicate with their environment. It affects the organization of other things at the cell surface. It affects so much, and these proteins are important, and we've known they've existed for a long time, but they're incredibly hard to study because of these dense regions of modifications. And so one of the things that Carolyn's work and many others in the field have done is to be able to bring bioorthogonal chemistry to mucin so that we can grab onto mucin domains and study where the glycans are in a site-specific manner, and understand what they're reacting with and how they're influencing the cell surface environment. That's what a huge push in Carolyn's lab has been recently, especially with respect to mucin domain glycoproteins. And these are upregulated in cancers, and they're important even in virology. We've seen several studies with SARS-CoV-2 and how mucins affect its ability to bind, and all these different things are interesting from a cell surface perspective. And so that's kind of the work that I do. And so mucins are hard and challenging. We're developing new tools, but I think that's just one section of the lab. What I really appreciate, I can't remember who mentioned it earlier, but Carolyn has touched on so many different aspects of biological need and chemical biology. So we've done tuberculosis work, we do cancer work, we do virology. So it's just all over the map of how we can apply these really creative ideas to study biological reactions in living systems, or chemical reactions in living systems. One other thing I'll mention when people are asking about application to cancer, right now, a really exciting application that Carolyn worked on in the mid-2010s was using sialidases, which are these enzymes that chew off certain sugars on the cell surface. And those sugars are important for engaging your immune system. And so basically, if your immune system sees these sugars, they stop a reaction of it quelling down your immune response. And so if you have a cancer cell that has a lot of these sugars on its cell surface, your immune system won't recognize it as a bad thing and won't act on it. And so what Carolyn has done is used antibodies in the field that already target cancer cells, used bioorthogonal chemistry to attach these enzymes called sialidases to an antibody that brings them to a cancer cells and removes this sugar from the cell surface. And then it kind of exposes it to the immune system so that your immune system can do what it's designed to do. And so it's ideas like that where you can see there's already like 3 fields of ideas that are coming together at a nexus that Carolyn's been able to kind of lasso together and have clarity on how to attack a really challenging problem in cancer biology with some really creative chemical tools.
Wow, that's absolutely amazing. You obviously explained that you came in from a mass spectrometry side. Did you always know you wanted to work with Carolyn? Because it sounds like her reputation precedes her, and it's like I think she sounds like an amazing person to work with. But had you heard of her when you were doing your undergrad or anything?
Yes, so I hadn't heard of her in undergrad, to be honest, but in grad school I heard about her early on. And in my mass spec training, I was absolutely a tool builder. I worked on instrumentation and how to fragment biomolecules in the gas phase. So I was always a tinkerer. And then I kind of learned about some of the reactions I was doing were particularly useful for challenging biomolecules like glycans. And I didn't know what a glycan was. I was like, well, okay, that's cool. Maybe we can use it for this. And immediately digging into what applications we could explore, Carolyn's name rose to the top. And so I was aware of her from early on, and her impact is, I'd say, from someone not in the chemical biology field at that time, her impact was almost greater outside of her science and her mentorship and her advocacy. And that's kind of where I saw like, oh man, this woman is really just has a platform that she's making sure to advance all the people around her. And so that was an exciting thing to see. And then you learn about her science and you're like, oh man, this woman is amazing. So like, I want to know more about her. She came and gave a talk at University of Wisconsin where I trained as a graduate student. And I basically, the person who was organizing the student lunch during her day visiting, I was like, please get me in that room. I think I want to postdoc with Carolyn and I'd love the ability to just connect with her. And so luckily I was able to get in the lunch with 45 other students, and I did everything I could to just ask a question so that later on I could email her like, hey, I was that random guy that asked you this question, and there was a great connection point, and she responded to me and we had a great conversation, and that's kind of launched my interest into being a postdoc in her lab. And so the personableness and the ability to connect with students and the willingness to engage with students in a lunch, remember who asked certain questions, and then follow up with me after email, just again speaks to kind of the character that she brings to her science too.
Yeah, and I've been in that situation during my studies and postdocs, and it's very scary to go up to those senior scientists that you respect so much and ask them those questions. And I think do you have any advice for kind of early career scientists now who are in that position thinking, oh, I'm setting out into the world of science, how do I approach these people? Because they seem like, so scary, but actually when you go and meet them, it turns out most of the time they're lovely people. So yeah, what's your advice for people setting out maybe in this field or in other fields right now?
Absolutely. Well, I would be first to say my advice is not the only advice you should listen to. There's so many great people I think could give, even on this call, that could give wonderful advice too. But for me, I've never regretted asking someone a question or going up and introducing myself, but I have regretted not doing that. And so to me, taking advantage of being at a conference with someone or being at a seminar where someone's giving us a talk, we all in this field, we like connecting with people and talking about our science and learning how our science can connect with another person's science. And so I'd say the main takeaways, don't be shy or be afraid. Know that a person will be more excited that you said, hello, I think your science is great, I'd love to talk to you more, than if you just walked out of the room after the seminar or something like that. So my advice is, work up all the the courage you can and go talk to the people, because they'll be very happy that you did as well.
And it sounds like it's a field that, you know, is so diverse that you've got scientists coming in from all different areas that actually no one's the specialist, right? You've got so many different specialists coming in that actually you need to communicate with each other and not be embarrassed if you don't know all the answers, because nobody knows all the answers. Thank you so much. That's amazing.
Oh yeah, sorry. Well, yeah, I'll just say I think that
Carry on.
speaks to the application of the click chemistry and bioorthogonal chemistry, is that I think MG was saying it brings in new ideas because of this platform it now has with the Nobel, but even before that, we have new ideas constantly flooding into how we can use these reactions, and that is, I think, just a phenomenal, reason to celebrate this type of chemistry.
Completely. I'm going to go to some of the many questions we've got coming in from the audience, and I'm going to start with a really nice one to start with. I'm guessing this is from a teacher called Louise, and they're asking how can they best communicate this amazing chemistry to their high school classes. Now, I don't imagine this is something any of you learned about in high school, but sure enough, you know, it sounds like it could be easy to teach. So what would be the best way to engage students with this science? And this is open to any of you on the panel now. Just jump in.
I mean, I do teach this chemistry to our final year undergraduates, and I've taught a course for 10 years now called Chemical Biology Approaches to Drug Design, because it's so fundamental and useful across the whole of medicinal chemistry. I'm not sure that high school students are going to understand the chemical reactions behind it necessarily. You know, we're talking cycloadditions and things like that. That might be a step beyond what most of them have seen, but I think, you know, a lot's been said, you know, in the past few days about it being like a Lego-like reaction, and I think that's quite true in a lot of ways. You can think of the chemical components as being Lego components, and you know, you can mix and match those components. That's exactly why we used it in the first place. Some of the original click chemistry is because it was just so easy to take one partner and put something onto it, or put something completely different on, and it both worked, you know. So yeah, just like clicking a red piece of Lego or a yellow piece of Lego on, it didn't really matter which one you chose. It coupled together beautifully. So I think, you know, at that level, it's certainly possible to teach.
I've got an idea.
Some Lego bricks. Go on, MG.
Yes, so this is something I've done with 4th and 5th graders. It can be a little dangerous, so you got to watch out, teach, but have them all close their eyes or blindfold them, stand up in the middle of the room with no desks, put a big piece of paper on one of them, only one of them, maybe a piece of newspaper, and have them all close their eyes and walk around very carefully and slowly with their hands out. And the idea is to have the one person find the person with the piece of newspaper on their chest, where they're just bouncing around, they can't see, and the idea is when you find that piece of newspaper, you've found your partner, you can make a bond, you can make a connection. It's really fun, and it teaches them a lot of things about chemistry, but also a lot about selective chemistry, right? I'm only looking for the one with the piece of paper on it, and I can find them after just a couple of minutes, actually. It doesn't take very long. As long as you avoid concussions and you know, people running, little boys running around is the you know, the devil in this one. But you know, that kind of thing, because you know, selectivity, connectivity is really easy to understand, and that's the whole power.
That does sound fun. I'm hoping Louise feeds back to us when she's tried this technique.
Don't
and maybe
sue me.
wins Teacher of the Year. Yes, I'm not interested in the health and safety forms. I want to know what happens. That's amazing.
Yeah,
I would love to.
I was just going to say, I think the thing that captures is it's about this concept, and it goes back to the kind of root concept of click chemistry. It's about connection. It's about building things in a really efficient way, and everybody can understand. I think, you know, even high school students hopefully still do some chemistry practicals. So they've done a little bit of boiling stuff up in flasks, and they've seen a little bit, hopefully, of the kind of the messy, smelly side of chemistry. And so if you kind of go, well, this is a bit like that, but it's really clean and it's really easy and it just works every time. That's I think the level that you kind of get to. But it's a long time since I was high schools. Yeah.
Well, MG, I don't know if you were part of the analogy. I've heard Barry, like, and even with the Nobel Prize this week, commenting like the seatbelt buckle analogy, and like you have one side that buckles into another, and that ability to connect things in a specific way is, I always thought, a great way to describe click chemistry. And so that could be another analogy. It doesn't matter what either side of the, you know, the belt is attached to, but if you can click these things together at their buckle, then that is a great way to connect molecules together.
That actually was Barry's imagery and inspiration, and in fact, that's where the term click chemistry comes from, is that satisfying click buckle sensation. So yeah,
there you go.
Didn't Derek Lowe use Velcro as an analogy this week? So yeah, it works beautifully. You know, it really connects two sides together, but you don't see it. You just see the two things that it's bringing together, but it's seamless.
The other concept that high school students, I think, might appreciate, and this was explicitly, we took explicit inspiration from this in the first click chemistry paper, was the power of connecting small building blocks to make sophisticated, bigger structures. So the analogy, of course, directly is to proteins and polymers. High school students know about that. And so a conceptual connection to connecting small molecular building blocks that are not necessarily natural ones to make a larger molecule that has a more sophisticated function and structure is again, maybe something they can grasp.
Wow, that's so many ideas. Thank you so much, all of you. We've all had someone giving us an idea there, which is brilliant. I'm going to get back to these questions because there's so many coming in, and we're going to dive into a bit more of the chemistry here. So I've got a question from Amir. What are the most important products formed in click reactions, or is there one product that is the most important? I mean, that's probably quite a hard question to answer, but anyone who wants to dive in with that one.
I guess I can start. So the prize highlighted the azide-alkyne cycloaddition reaction. So the obvious answer is the triazole, that 5-membered ring, the 1,4-triazole. But if you broaden your view to look at the larger world of click reactions, their connections are very varied. But again, the interesting one is there are very few carbon-carbon bond-forming reactions. There are in the cycloaddition case that aren't cycloadditions. There are very few carbon-carbon bond-forming reactions. And the reason for that is that carbon-carbon bond-forming reactions tend not to be strongly thermodynamically driven and have a selective reaction path. Aldol condensations, you know, the quintessential one, they can go in a million different directions. And so what tends to happen is you make bonds between preformed units that have a lot of carbon in them. And Barry is fond of talking about nitrogen as the key connector with azides, but also in later years, sulfur connections to oxygen and nitrogen have been important. But if you had to point to one, it would be the triazole. Very stable, dipole moment like a transamide bond, structure like a transamide bond. So it's really, again, biologically quite friendly.
I'm getting a nod from Alison there, I think, yeah.
That's the one that I highlight in my lecture course, so yes,
Okay.
I'd have to go with that too.
Brilliant, there we go, we've got some agreement, I like that. We've got a question from Caviarati about, can you tell them a little bit about the reagents used in click chemistry and if, you know, it's the same reagents used in the click and the bioorthogonal chemistry? Does that make sense to you, to somebody?
Nick, do you want to kick off on bioorthogonal?
Sure, yes. So the idea that was the transition from the original click to the bioorthogonal is the idea of not needing a metal catalyst, so that can be done in living systems where that might be a complication. And so in the bioorthogonal chemistry that Carolyn promoted, this idea of strain promotion. And so here you have these same functional groups that work in click chemistry, the alkynes and the azides, but now you've put strain around them in rings. So you have carbon rings that are upping their internal energy and willingness to react at a certain speed. And so those molecules are kind of the bread and butter of some of Carolyn's bioorthogonal work, is in the sense that you have your, let's say, azide that you've incorporated into a biomolecule, and now you have a fluorophore that has a strained alkyne next to it. So you can do this reaction to connect them without needing the copper catalyst to drive that. And so you see things like, I'm going to forget what the acronym stands for, but DBCO is, I can't remember, there's basically a strained cyclooctane with 2 other carbons on the other side of it. So you can see there's all this other chemistry that's forcing this reactivity of the alkyne that's in this type of molecule that drives reaction. So those are some of the reagents you can imagine, but I'll say that the azide that you might imagine coupling in this can be on anything you like. We use it a lot for glycans, as we know about Carolyn's glycobiology. So we have monosaccharide precursors that have an azide stuck on them. We feed them to cells in a metabolic fashion, and they incorporate them into their cell surface glycan. So it's just really a UDP sugar analog, and you just put it in your cell culture, and it appears on your cell surface because of the natural building blocks of how glycans are made. And then you can add this strain-promoted reaction and do it in a live system. So it's the reagents are, just describing one of them is, that's such a narrow view. There's just infinite numbers of ways you can imagine having these types of reagents built and used.
And I think that's actually one of the interesting things of the whole field of bioorthogonal chemistry is the development of the new types of reagents, because we've not really got the ideal water solubility or maybe stability in this particular situation, and so actually there's a lot of things still left to actually optimize in a lot of ways in order for different situations. Do you want that to be cell permeable? Do you want it to, or do you just need to react with the cell surface? So all these sorts of things are important. Do we want a sulfate on there, or no, that's not any good, do we? So there are lots of different ways in which we can imagine functionalizing these reagents to actually improve them, and that's definitely something that's very active still at the moment.
So there's still much to do. This is, much for future scientists to be looking at. I'd love to come now to our second poll, actually. So if we can launch the poll, because I just want to kind of touch on a slightly different subject, moving slightly away from the science. But yes, I don't know, can we bring the poll up now? There we go. And it's basically a little fun one. Basically, how many women have won the chemistry Nobel Prize to date? If you want to just have a go, please don't Google it. You probably know the answer. Particularly if you've been reading our Chemistry World content over the last few days, then you probably will know the answer. But a little bit of fun. We've got quite a few people having a go at voting here. And I wanted to touch on this subject because I will give you a hint. It's not very many to date, as you might have guessed. And actually the poll so far is sort of reflecting that. But some of you are more negative than I was thinking. It's not quite as bad as some of you are thinking. But yeah, I wanted to touch on this. I think actually, could we bring up, I think we've had quite a few people vote now. If we could bring up the result of what people think. There we go. So 47% said 8, and they would be correct. I love that some of you thought 26. Unfortunately, that is not the case. But in terms of Nobel Prizes in general, I think, Philip, have you got any insight on this? Maybe it was about 60 women who have won Nobels in total. Is that right?
I can't. To be honest, I haven't got the numbers for total Nobel Prizes, but of those 8 women who've won chemistry prizes, 4 of those are in the last 5 years as well. So if you look at the whole history of chemistry from 1901 onwards, before 5 years ago, there were 4 women. And then there's 2009, and then you go back to 1965, and then you go back to the early 1900s when it's Marie Curie and her daughter Irene Joliot-Curie. So before that, it's like the early history of the Nobels is very, very, very missing in women, and we know that there were women doing fantastic work at that time, and they're just not recognized. And part of that, I think, comes from the nominations process. Katrina in our team and a couple of the other team members have been looking back at the nominations data, which is only released 50 years after the event. So we can look up to 1970. We can look from 1900 to 1970. You look at that data, there are only 67 nominations of women in a total of 4,300 nominations, which is crazy. And of those, there's 13 individuals, 3 of them won the prize. So hopefully we can see that ratio is changing mainly. In recent years, the Nobel Foundation has kind of hinted that it's making more efforts to encourage nominators to nominate more women, to think a little bit more about those things. But it's the kind of thing that's going to take a lot of time to change.
And it's not just a gender issue, it's all of diversity, isn't it? I was just going to ask May, actually, is that data you keep in terms of, your journal articles, in terms of first author, last author, has that been changing over the years? Do you think things are changing? I'm pleased to be positive.
They are changing, yes, but it is slow. I mean, to give you a sense of number of female authors, for example, we think about 36% of our authors in across all RSC journal articles are female. So we tend to take that as a bit of a benchmark of being reflective of the community that are publishing. Obviously, it would be great to see that at 50-50, but you know, let's start at 36% and see if we can actually be reflective of the community. And I have to agree with Philip, when we look at sort of journal awards and prizes as well, it comes down to the nominations, and we have to work really hard to remind people that actually, you know, got to nominate those brilliant women who are doing great chemistry, and and women need to make nominations. You know, I think we have a responsibility to make sure that voice gets heard.
I can see Alison wanting to come
I
in here.
know,
Please
I need to chip in here.
Please do.
So, you know, as president of the Organic Division Council from 2016 to 2019, the stat that the RSC will let me say is that I achieved a 420% increase in the number of female nominations. So that tells you that there weren't very many to start with. And what I think is really, really important for actually overall really, really important is there's a wonderful Harvard Business School piece in 2016, and that analyzes the number of a minority. So we're not, you know, this could be women, or this could be ethnic minority, and they looked at a couple of different situations, but one of the situations that they looked at is if you have 4 nominees for a position or whatever your interview is, and one of them is a woman, or is your minority, that minority has a 0% chance of being appointed / awarded or whatever. And I think what's really important is that we have more than one, because once you get to a 50-50, you're on an equal footing. So, but the problem is that if you are perceived as the outlier, the outlier has no chance of being awarded or considered equally. And so it's really, really important that we, you know, that we get more women on the list. So that was like one of my big things as Organic Division president, and it's one of the reasons why I'm so delighted that Carolyn's, you know, been nominated, awarded the Nobel Prize, because I mean, I've known her for a lot of years, and you know, and it's just really important, as I say, that we get more women on the nominations list, and more of all minorities on the nominations list.
Yeah, definitely. That's well, you know, there's work to do, definitely, but I do hope things are changing. But yes, I know it can be a hard time for women in science. So to see this prize, you know, go to the 3 of them is brilliant, and to definitely have Carolyn in there is really fantastic. We are nearing the end of our webinar. I can't believe it's flown by. We've had, you know, such a great chat here, but I would like to throw in a final poll, which is another sort of fun one. That seemed like a fun one, but we got quite serious after the last one. So if we can launch the final poll. The question is, Carolyn was in a band called Board of Education with Tom Morello, and we're just wondering if you know which band Tom later gained international acclaim with. I'm sure many of you know. I've actually seen one of the comments, somebody definitely knows the answer to this one. So it's a little bit of a fun one, and you know, I think Nick, you obviously know Carolyn. Does she still play in a band?
I haven't seen her actually play any instruments that she had in her office, but she used to have a bass guitar in her office. That I was a former bass player in another life, so I saw that when I was interviewing. I was like, this is amazing. But I have seen her play piano. She's a great pianist as well.
You know, I can't believe she has the time to play instruments as well as all this amazing work she's doing. But okay, so let's see. Quite a few people have voted. Kings of Leon and Metallica are quite popular. Rage Against the Machine is very popular. I think we'll just we can release the results now, because I think, you know, this was just a bit of fun. And those that said, which is about half of you said Rage Against the Machine, and you would be correct. Yeah, I think that's amazing. I did see something on Twitter from Tom Morello that, yeah, I think Nick, you put this in an email to me earlier, didn't you? About him saying that he was, you know, more excited to know her now than she was to know him.
That's right. Yeah. So now she used to say, I'm in a band with Tom, but now Tom gets to say, I was in a band with Carolyn, which is great.
I know. I love that. It's very cool. Like, yeah, I saw as soon as it was announced, he was straight on Twitter. So that was fantastic. So look, thank you to all our wonderful guests for joining us today. It has been a brilliant celebration of the Nobel winning scientists and the science. And, you know, thanks to all of you at home and wherever you are in the world, our audience, for getting involved in this conversation and asking some brilliant questions. Now, of course, we'd like to thank our sponsors again for enabling us to make this webinar happen. One of them is Shimadzu, who are involved in the manufacturing of precision analytical instrumentation. And the other is Jump, who are a data analysis tool of choice for hundreds of thousands of scientists, engineers, and other data explorers worldwide. Now, a link of today's recording will be sent out to everyone who registered, and we'll also be uploading the recording to YouTube, so you can share it with your friends and colleagues if you think they would be interested in learning more about this subject. If you've enjoyed this webinar, then please check out chemistryworld.com/webinars for future events. And whilst you're there, do feel free to register for a Chemistry World account, where you can sign up to our very popular Reaction newsletter that includes a weekly digest of big stories from across the world of chemistry. I'm Natalie Starkey, Chemistry World Science Media Producer, and I look forward to seeing you again in a future Chemistry World webinar.